Hi, my name is Keahi, and this past year I've been researching the difference in probiotic bacterial content of poi from various production methods. So being from the native Hawaiian ancestry, I was interested in preserving the growth of taro and production of poi on the Hawaiian Islands. When I found out that poi was a potential probiotic, I was curious to find if it actually contained the bacteria that benefited one's health. Also, I was interested in the health of children, and I'm aware that most infants are highly allergic to dairy and dairy products, and poi being hypoallergenic, it can prove as a great alternative for these products, and so I was curious to really find out what bacteria this product contained. So regarding poi studies, I found that poi is a staple in Hawaiian culture and is highly nutritious. However, there's little known about the bacteria that influences its ability to produce health benefits for the consumer. So in my initial research, I found that probiotics are live microorganisms that transit the gastrointestinal tract and when consumed confer a health benefit to the host. So Lactobacillus acidophilus is the leading lactic acid producing bacteria located in probiotics such as yogurt and produces a variety of health benefits. Beneficial effects of probiotic bacteria include enhancement of the immune system and reduction of lactose intolerance. Therapeutic applications include prevention of urogenital infection and alleviation of constipation. Because the lactobacillus bacteria is found in yogurt, I hypothesize that it will also be the most prevalent bacteria in poi as well, due to its similarities in consistency and its use of the same fermentation process. Another common probiotic, the Weissella genus, consists of bacteria that have been isolated in the breast milk of mothers, milk of animals, and is often found in fermented foods. Specific Weissella strains are also receiving attention as potential probiotics, so I was looking at this lactic acid producing bacteria as well. Many factors also influence the viability of the probiotic bacteria, including fermentation, pH and acidity levels, and storage and incubation temperature, as well as dissolved oxygen content. Yogurt and probiotic fermented milk should contain at least 1 million viable cells per gram at the time of consumption. And when using the culture-dependent methods, probiotics such as fermented milk have a higher stability during using cold storage. So my research question was, does the bacterial community and the amount of lactic acid producing bacteria differ between poi of distinctive production methods? And going back to the numerous papers I looked on this research, I found that probiotic bacteria is often found in yogurt, and I found a lot of health benefits from these probiotics. And I also found that there were differences in the production and storage conditions, and that leads to differences in the probiotic bacterial species and amount. So the purpose of my research and the gap between the research that was already conducted by others was to identify the species and count the amount of colonies of probiotic bacteria in poi, while also comparing it to the processes used on yogurt and looking at the production methods on the viability of probiotic bacteria in poi. So for my experimental process, I looked at 3 different samples of probiotic products. The first sample of poi sample 1 being kept refrigerated immediately following production until being shipped out to the markets where it was kept at room temperature. Poi sample 2 is pasteurized and kept refrigerated from start of production to consumption. And the yogurt, which went through homogenization, pasteurization, and then fermentation. After these products, after I sampled some of the product, I diluted it in peptone water to obtain a countable amount of colonies. So first, I took the diluted products and spread it on specifically MRS agar with a sterilized spreading tool, and then placed these plates into gas packs that acted as an anaerobic environment through incubation. Then I picked the colonies and extracted the DNA by heating the samples, spinning them down, and pipetting the top of the liquid off. And then using this DNA supernatant, I added it to 16S primers and the TAC master mix to amplify it through PCR. Finally, these samples were sent off to Laragen for sequencing to obtain nucleotide sequences. So for my results, I created this nucleotide alignment. And do you guys recall the nucleotides A, T, G, and C from biology of DNA sequences? Are these 9th grade biology memories sinking in? This is a nucleotide alignment of sequences from the BLAST database. So if you see how the first sequence has the C nucleotide, while, for example, the Y confusa sequences have T's. So it compares the different sequences that I obtained and comparing it to the reference sequence from the database. And so the purpose of this alignment is to compare the sequences, and it shows that POI sample 1 matched 100% to Y confusa, which is also a lactic acid producing bacteria. And although I hypothesized that it would match to the Lactobacillus acidophilus species, it instead matched to a different lactic acid bacteria. POI sample 2 actually contains no colonies that formed on specifically the MRS agar, and the yogurt correlated to Lactobacillus rhamnosus 100% with its nucleotides. And so POI sample 1 contained the most probiotic bacteria, as I found through the different the colony amount on the plates. And POI sample 2 contained no bacteria on the plates, and then yogurt contained some, but much less than POI sample 1. So POI sample 1 contained over 35 million colony-forming units per gram, and CFU, or colony-forming units, estimate the number of viable bacteria in a sample. The bacterial counts in POI sample 1 correlate to the bacterial count of dairy products, which is around 10 to the 7th or 10 to the 9th colony-forming units per milliliter, and these probiotic yogurts contain bacteria that are able to stimulate the gut microflora and claim specific health benefits that I mentioned earlier. Also, the National Yogurt Association introduced the live and active culture seal, which states that these products should contain at least 100 million live cultures per gram. And therefore, by containing a bacterial count in the range of milk-containing products and one that has a much more than 100 million colony-forming units per gram, sample 1 is significantly healthier than the tested yogurt and POI sample 2, and is considered probiotic. In addition, POI sample 2, having not contained any products, was due to it being refrigerated and pasteurized, and is not a probiotic. This also shows that production and storage methods definitely matter in identifying the species and amount of probiotic bacteria. So not only does POI have health benefits, but it also can be used as an alternative for dairy products since it is hypoallergenic, which is why POI is often fed to infants. POI consumption has unfortunately plummeted in these recent years and has taken a toll on taro production as well, and therefore the preservation of many Hawaiian traditions. Greater knowledge of bacteria in POI and taro and its benefits can lead to a possible increase in taro production and in the Hawaiian traditions as well. So there were many fascinating questions that came up during my research, including that because I only came up with 3 different tests for these different samples, in order to generalize and validate my results, I would like to test a larger sample size of the POI and possibly sequence all of the colonies that were on the agar dishes to obtain all of the probiotic bacteria that could be in the POI. And this would test the morphologically different or similar species that were on the agar dishes. And I purposely chose different batches to obtain this overall probiotic bacterial count, but I believe that by testing all of the colonies that formed, it would be more general. And however, I would also like to test the shelf life prior to consumption and to see, for example, if it's sat for more days than another POI, then it would contain more or less probiotic bacteria or different species. And other than a larger sample size and different shelf life, I would also like to test different varieties of taro and the traditionally pounded POI versus store-bought POI. So throughout this process, I learned that the experimental trials and data analysis is quite time-consuming because there are many factors in this process and results that are often not expected. And however, when you conduct research that really interests you, like how mine did, it becomes so worth it in the end. And all of the data that you collect and the data that's analyzed is really valued to you and to the community around you, which is really cool. Also, it has been an incredible experience to obtain lab and data analysis skills that can definitely help in the future. So I definitely recommend this class to anyone thinking of taking it. It's great. And so I would like to thank Dr. Chen, especially, for being my overall mentor and teacher. And I would also like to thank Tiana Abley, who helped me learn about the different production and storage methods, and HPC, and Dr. Chin Lee and Dr. Yang Lee, who helped me learn more and figure out what I wanted to do for my future research, and Ms. Fingston for helping me realize the importance of poi and taro in Hawaiian history. Thank you.